This is part 2 of 2 on chapter 10, Genetic Engineering. In the previous part of this chapter, we looked at very briefly different tools and techniques of genetic engineering. Now we're going to take those different tools and techniques and kind of put them together to see different possibilities that we can do, or how we can apply these different tools and techniques. So we're going to look at this question: how are these different tools and techniques used in this applied science? So one thing that we can do in genetic engineering is looking at recombinant DNA technology. So recombinant DNA technology, this is where we have an intentional removal of genetic material from one organism, and we're going to put it into a different organism, or combining it with a different organism. So the object of this recombinant technology is cloning. So taking a gene or DNA from one organism and cloning it into another organism. So I'm going to kind of run through this recombinant DNA technology. So we can take a eukaryotic cell or any other type of cell. It can be human, other mammal, microorganism, or plant cell. In that cell of interest, we're going to isolate our piece of DNA that we're interested in. And we're going to make that DNA from an mRNA. So we're going to use a reverse transcriptase enzyme that we looked at in the first part of this chapter to take our mRNA and create a cDNA molecule. And we want that cDNA molecule because we only want the genetic part of the gene that's actually expressed. We don't want those introns in there. So we have this clean piece of cDNA. We're going to take that DNA and insert it into something called a vector. And a vector is usually a very small piece of DNA in the form of a plasmid or in the form of a circle. So we take our blue DNA that we're interested in and insert it into this pink vector. This vector, we then put it on a plate with bacteria. So usually we use different bacteria species as our cloning host. So we have a microbial cloning host. We're going to transform it. So these bacteria cells are going to pick up this vector from the environment and put that foreign DNA into the microorganism. So now our vector, our little piece of DNA that has our DNA of interest in it, is now inside of our cloning host. That cloning host then goes through binary fission, so that bacteria cell is going to replicate itself. So it's going to clone or amplify that gene of interest we're in. And once your bacteria clones itself, the cell can actually express that piece of DNA. So it can, for example, on the left-hand side, we can have that bacteria cell produce a protein product. This is a lot of pharmaceuticals, hormones like insulin, human growth hormones. These bacteria cells can produce different vaccines or industrial chemicals. So they can actually produce our molecule that we're interested in. Another thing that you can do with this cloning host is that you can take that cloning host and put it into other organisms. So you can alter organisms for economically useful traits. So this is in the middle. And we can create plants that are pesticide, herbicide resistant. We can improve the nutritional value of different plants. So that's one economical application to this process. Another one is multiple applications in genome research and testing. So we can figure out gene function, we can sequence that gene, can identify, we can make gene libraries just for more knowledge, for more understanding. So this process is very useful. We can actually get pharmaceutical drugs from it, or we can alter organisms or use it for research. So characters of these cloning vectors. So the vector, it must be able to carry a significant piece of donor DNA. So a vector, remember, is that plasmid, the round piece of DNA. So the vector has to be able to carry our DNA of interest, and it has to be readily accepted by the cloning host. So our cloning host has to accept the vector, and it has to be able to live with the vector in it and produce that gene of interest. So usually for vectors, we use what are called plasmids. They're small, well characterized, easy to manipulate, so we know what's on these plasmids, and they can be transferred into the host cell through transformation very, very easily. Another type of vector is called a bacteriophage. So remember, these are bacteria viruses. They have a natural ability to inject their DNA into bacteria hosts through transduction. So plasmids, bacteria or the cloning host pick it up through transformation. They just pick it up through the environment. Bacteriophages, this is a type of transduction where we put that piece of DNA into the virus injects the DNA into the cloning host. So it might be good to go back and review conjugation, transformation, and transduction that we talked about in Chapter 9. And that'll help you kind of understand these two different types of vectors better. So vector considerations, again, we want a vector that's really well characterized, we know what's on it. We have to know the origin of replication is needed, so we can actually replicate the vector. So it has to have an origin of replication. The vector must be able to accept DNA of a desired size, so the vector has to hold our gene of interest. And the gene has to confer drug resistance to their cloning host. So the drug resistance, this is how we're going to identify if our DNA piece that we're interested in has been put into the vector. Alright, so we looked at the vector, that's the piece of DNA that's going to hold our gene of interest. We had to put this vector into a cloning host. And usually we use microbial cloning hosts because they replicate very quickly. So we want a host that is going to do rapid turnover, fast growth rate. The microbe can be grown in large quantities using just ordinary culture methods in the lab. We want one that's non-pathogenic, so we don't want to get our scientists sick. The genome has to be well mapped, so we know where our vector is in the genome of our host. And we have to have something that is actually capable of accepting the plasmid or bacteriophage vector. So it doesn't help if we can't get our vector into our host. And our host has to maintain these foreign genes through multiple generations, so they have to be pretty stable organisms. And the cloning host has to secrete high yields of protein from the expressed foreign genes. So good cloning host, a really good one is E. coli. E. coli, it's a little bit pathogenic, but usually it takes a lot of E. coli to get someone sick. E. coli can go through binary fission every 20 minutes, so it grows very, very quickly. So E. coli is a very good cloning host. Okay, so different products that we can get from this recombinant DNA technology, so taking a gene of interest and putting it into another organism, we can enable very large-scale manufacturing of life-saving hormones, enzymes, or vaccines. So insulin is a really good example of this. We looked at the human growth hormone for dwarfism, so we can have a lot of this human growth hormone produced in the bacteria, you collect it, and then you can use it to treat people. Erythropoietin for anemia, and then there's vaccines we can get, or other drugs, or hormones that can be produced using this technology. Another part of this is we can create genetically modified organisms, or GMOs. They're also called transgenic organisms, so different terms. Usually in the media they use GMO, or genetically modified organisms, because it's a little easier to understand than if you say transgenic. That one you'd have to actually know what it means. So we have created recombinant microbes. So there's different bacteria. So Pseudomonas syringae prevents ice crystals in different liquids, so we can put this bacteria in there to prevent the liquid from freezing or forming ice. Bacillus thuringiensis, hope I'm saying that right. This encodes for an insecticide, so we can put this bacteria into different plants, and then that plant can produce their own insecticide. So many enzymes, hormones, and antibodies used in drug therapies are manufactured using mammalian cell culture. So this is another genetically modified organism we can use. So we can take mammalian cells, we can modify them, and then they can also produce hormones or different drugs. And cell cultures, really cool thing about them is they can also modify the proteins. So the proteins are just not made. The cells are eukaryotic cells. They can modify these proteins. They can do a little bit more with the drug that we're producing. So in order to make transgenic plants, we just use the same type of method. So we isolate a gene that we're interested in. We put it into a plasmid, into our vector. We then put that vector into our cloning host. So this is the very top A and B part of this. So we have our cloning host all set up. This cloning host, we then infect a plant with the cloning host, and we hope that the plant then takes in our gene of interest and incorporates it into its own DNA. So this is how we can get a plant that can produce its own herbicides or pesticides. So we just have to go one step further. We have to take our cloning host and use it to infect our plant that we want to have this special property. So examples of transgenic plants, and you can look in your book as well. Let's see. So we can modify plants to have higher levels of antioxidants, so we can increase the nutrition. We can give the plants longer shelf life. We can allow the plants to produce their own pesticides or herbicides, improve nutrition. So those are examples of different things we can do to plants as well. A good one is golden rice. We've improved the nutrition on it so that we can give it to developing countries and help them with different deficiencies that they may have. Transgenic animals, usually we use a virus to infect a fertilized egg or early embryo to get our gene of interest into that organism. And then transgenic animals can transcribe and translate eukaryotic genes. So what we can do, we can take our gene of interest, put it into a virus, into that we then infect that organism with the virus, allow that organism to grow and develop, and then we have transgenic animals that can be produced. And there's a table in your book looking at different transgenic animals. So we can use animals for something called farming. So farming with a PH, it's a combination of farming and pharmaceuticals. So you can have different pigs, sheep, cows that produce different drugs for us. We can also do other things. One example, we can make cats and dogs that are hyperallergenic so that people that have allergies can still have pets. Other examples, we can create glowing organisms, and that's usually more to study embryology or how organisms develop from an egg until they're an adult. There's glowfish. I believe you can buy those from Walmart. I think they're still kind of expensive, but you can see the fish up at the top right. We have growing different organs on mice. So on the bottom right, there's a mouse that has an ear growing on it. So you can grow different parts of humans on animals. So there's lots of different applications to this idea that we're looking at. We can also use something called a genetic treatment, so we can introduce DNA into the body, and this happens during childhood or as an adult. So you can do things like gene therapy, where we can correct or repair a faulty gene inside of an individual human. And there's two different types of gene therapy. There's ex vivo. This is the normal gene as clone and vectors. Tissue is removed from the patient. That tissue is infected with the vector, And then once the tissue starts to grow and has the normal gene, we put it back in the human. So ex vivo therapy, your patient tissue is infected outside of the body. In vivo therapy is where you just take your naked DNA or vector and you directly put it into the patient. And this is shown in the image here. So we take our DNA up at the top, we replicate it, we put it into a virus, and then we infect that individual with that virus that contains our gene we're interested in repairing or correcting. So this is ex vivo means outside of the body, in vivo means inside of the body. Another application to the different tools and techniques we've looked at is called genome analysis. So we can do DNA fingerprinting. So every individual has their unique sequence of DNA. It's kind of like your fingerprints on your hand. You all have unique DNA, so DNA fingerprinting. So the methods that are used in this include restriction endonucleases, so we can cut up pieces of DNA, electrophoresis, separate that DNA out by different size. We can hybridize the DNA, or we can do something called a southern blot, where we can identify the DNA and the proteins that are being produced. So this is an example up here for DNA fingerprinting. So in this example we have, I think it's a case of rape. Yep. So we have a victim that was raped, evidence collected off the victim, and then we have two suspects. So you take DNA from the suspects, DNA from the victim, DNA from the evidence, you cut it up into little pieces, run it down a gel, so separate it out by size. Each individual human, they're going to end up with a specific banding pattern when you do this process. You visualize that banding pattern, and then you can compare your victim and the evidence to your suspects, and you can actually identify if a suspect was at the crime scene. So on the bottom of this image, where it says forensic test, if you look really carefully at the bottom, you have your victim. You have evidence collected off the victim, evidence 1, evidence 2, and both pieces of evidence have the same banding pattern that you can see on the image. And then up towards the top you have suspect 1 and suspect 2. And you'd compare those suspects to the evidence collected off of your victim. And you can see suspect 1 matches the evidence that was collected off the victim. So they can actually use this in court to say that suspect 1 matches the evidence that was found on the victim. Therefore, we think this suspect was the one that did the crime. Right here. So very powerful tool for this, the DNA fingerprinting. So this DNA fingerprinting, we can use it to identify, besides identifying criminals or victims, we can also use it to identify hereditary relationships, so looking at if two humans are related to each other, how closely they're related. We can study the inheritance of the patterns of disease, we can study human evolution or evolution of other organisms, and then of course identify victims or criminals. They can also use it to identify victims of different disasters, like 9/11. I believe they're still analyzing DNA pieces from that, but they're using this DNA fingerprinting to identify the victims and to help give closure to the families. We can also analyze the mitochondrial DNA to trace evolutionary relationships of different organisms.